Hi, I'm Dora. I work in the Pharmacology department at the University of Oxford, and I'm actually a British Heart Foundation funded research fellow, and I'm also an academic in the department as well. I work on the microcirculation, so the smallest blood vessels in your body. And I do a lot of imaging to look at those blood vessels, but I'm also now starting to do a lot of work with human tissue, including the blood vessels and the cardiac myocytes themselves, so truly cardiovascular physiology and pharmacology. We're very interested in how the endothelial cells that line the lumen of all blood vessels communicate with the surrounding smooth muscle to cause contraction or relaxation. And part of this process involves very rapid changes in calcium in the endothelial cells and in the smooth muscle cells. So we're very interested to image these events as fast as we possibly can and align them with as good temporal and spatial resolution as we can to certain structures in the wall of the arteries. So basically I need to be able to image multiple cells in one frame and at a rapid speed. So I'm using particularly the line scan confocal microscopes, and we have quite a few Olympus ones for that, but I also have a NIPCO spinning disc confocal microscope on an Olympus microscope. So the two gives us different benefits. So the spinning disc is very rapid, but the disadvantage of that is it's only a single wavelength for our particular system. So if we want to do multiple wavelengths, we move to the line scan confocals, and we quite often use fluorescent indicators for cell structures between cells and within cells, as well as doing calcium imaging. So the ability to have multiple laser lines to image at a fast speed is what we always will desire. And the bigger thing in our system is the cells are very delicate, so we want to use the minimum amount of laser power as possible and have a very sensitive detector system. So we've just moved to the gasp detectors that are available now for our microscopes. In all our experiments we do freshly isolated or dissected tissue. So either from animal models or from people, so from patients that are going bypass or cardiac surgery, we get samples of tissue. So it's really important that all of those cells remain as they would have been in the original situation. So we have to be very careful with how we prepare solutions and dissect. And then for the blood vessels, we actually isolate them and cannulate them at both ends, and they're very small and delicate, so it's quite fiddly work. But then once it's set up, we can image them for hours and hours, so we really like that. Quite a few microscopes, and they're all from Olympus. And the nice thing is that if we have a particular experiment that we want to do, but that rig isn't quite set up, we can always move, objectives or stages or other pieces of equipment from one rig to another and design each rig to particularly address a question. So that's very flexible. So it's very nice that we can move equipment between rigs. And then obviously having the ability to do the speed and the multiple wavelengths for us is always important.